Usually when we talk about getting a medical diagnosis, there's an expectation of almost, I don't know, engineering level precision.
Right, yeah. Like it's just binary.
Exactly. You break your arm, the x-ray shows that jagged white line, and the doctor just points to the screen and says, well, there it is.
Yeah, it's clean, it's visible, and frankly, it's comforting. I mean, we naturally prefer when our physical ailments can be just perfectly categorized.
Right, but then you step out of the orthopedics ward and, well, you go into the world of analyzing microscopic tissue samples and genetic expression, and suddenly that x-ray machine is just completely useless.
Oh, totally useless. You're looking at a diagnostic landscape that is incredibly murky.
Exactly. So, welcome to the deep dive. Today, our mission is to unpack this revolutionary new computational framework called CODA. That stands for Integrative Cross-Sample Alignment and Spatially Differential Gene Analysis.
Quite a mouthful, I know.
Yeah, it really is. And, to understand why CODA is sending shockwaves through the biological community, we first need to look at how we currently study genes. So let's unpack this. Imagine you want to know what exactly makes up a specific diseased tissue, say a tumor in a liver. The traditional method, RNA sequencing, is basically like taking that tissue, tossing it into a blender, and making a smoothie.
That is a highly accurate, if slightly graphic, analogy.
Right.
But if you analyze that biological smoothie, you can know exactly what ingredients or genes are in the mix. You know you have strawberries and bananas, but you have absolutely no idea how those fruits were arranged before you hit blend. You lose the entire original architecture of the tissue.
Exactly. You know what is there, but not where it is. And that's where spatial transcriptomics, or ST, comes in. If traditional sequencing is a smoothie, ST is like a highly detailed city map.
Yeah, that's a great way to put it.
It doesn't just tell you that you have muscle cells and immune cells. It shows you exactly which cells, think of them as houses, are expressing which genes in their exact physical locations.
And that spatial context is everything. I mean, it allows us to see how cells interact in their local microenvironments, like who is talking to whom, where are the immune cells gathering.
Right.
That geography is vital for understanding developmental biology, cancer, and just incredibly complex disease mechanisms.
Mm-hmm.
But well, we run into a massive bottleneck here.
We have incredible tools to create these molecular maps, right? Technologies with names like 10x Visium, MERFISH, and Stereo-seq.
We do, but the problem is that analyzing multiple tissue slices together is a logistical nightmare for researchers.
Because the process of making the map actually destroys the landscape, right? You can't slice a piece of tissue that thin without damaging it.
Precisely.
Yeah.
When scientists slice tissue incredibly thin to mount it on a glass slide, it gets stretched, torn, rotated, or, magnified differently during the lab processing.
So you end up with multiple maps of the exact same biological city, but one map is printed upside down, one is stretched out of proportion because someone pulled the tissue too hard, and one has massive microscopic tiers separating the neighborhoods.
Yeah, it's a mess. And if you want to compare a healthy slice to a disease slice, you have to align them perfectly. But traditional alignment methods either only do rigid rotations, like just spinning a piece of paper on a desk, or they require massive supercomputer-level processing power to calculate how to fix the complex distortions.
Which most labs don't have.
Exactly. Even worse, as we'll see, traditional analysis frequently misses crucial disease markers entirely because it's only counting the total number of genes, not looking at where they are located.
But by the end of this deep dive, you will understand exactly how CODA solves this massive biological jigsaw puzzle, why it's dramatically faster than its competitors, and how it uncovered hidden heart disease genes that other tools just completely missed.
It really is a paradigm shift in spatial biology. To understand how CODA finds these hidden disease markers, we first have to look at its engine. How does it act as a master cartographer to perfectly align those distorted maps?
Right.
It does this by moving from the macro level down to the micro level.
So getting the map right side up. If I hand CODA you two incredibly messy, distorted slices of tissue, what does it actually do first?
So CODA starts with global alignment. It takes the highly complex gene expression data from both slices and projects it into a shared low-dimensional space.
Meaning it kind of groups the data to find shared biological communities?
Exactly. It identifies where the major structures are. Then it estimates a global transformation using 3 different modes. Mode 1 is rigid.
Which is just rotation and translation, right?
Right, preserving the absolute physical scale. Then mode 2 is similarity, which adds uniform scaling.
Okay, to fix overall magnification differences if one slice was zoomed in more than the other.
Exactly.
Makes sense. Spin it and zoom it.
But mode 3 is where it gets sophisticated. It's called affine alignment.
Yeah.
Affine alignment is crucial because it accounts for anisotropic scaling and shear.
Okay, wait, let's pause on anisotropic scaling. I'm picturing silly putty. If I have a piece of silly putty with a picture printed on it and I pull it from all four corners equally, that's uniform scaling. But anisotropic scaling means I'm only pulling it from the top and bottom, stretching the image vertically while the horizontal stays the exact same.
That is exactly right. When tissue is cut and placed on a slide, it rarely stretches evenly in all directions. It shears and skews along specific axes.
Oh, because of how it's physically handled in the lab.
Yeah. The affine mode mathematically reverses that specific directional silly putty stretch so the global shapes match up again.
But wait, I have a logistical pushback here. What if the two slices don't even cover the same area to begin with? Let's go back to our city map analogy. What if slice A captured just the downtown area and slice B captured the entire county?
That happens a lot, actually.
Right, you can't just stretch downtown until it fits over the whole county. The biology wouldn't match at all.
This is exactly why many older spatial algorithms fail or, require scientists to manually crop the images beforehand. CODA automates this using a brilliant mechanism called common domain identification.
How does it do that?
Well, CODA renders the complex multidimensional genetic data into a multi-channel image. If we use 3 dimensions of data, you could literally think of it as converting the math into an RGB image, a standard red, green, blue color photograph.
Wait, really? It takes millions of spatial genetic data points and literally turns them into a picture.
Functionally, yes. And the reason this is so clever is that by converting it into an image format, CODA can borrow highly advanced image matching AI that already exists in the computer vision world.
Oh, wow.
Yeah, it uses a transformer-based AI model, similar to one called Light Glue, to visually match key points between the two slices.
So it looks for matching visual patterns in the gene expression.
Exactly. Once it finds those matching anchor points, it crops out a common domain. It isolates the overlapping region, so it is strictly comparing apples to apples, or downtown to downtown, in your analogy.
That is incredibly smart. Instead of trying to invent new spatial biology math from scratch, they just translated the biology into a format where existing computer vision AI could solve it.
It's a massive shortcut that saves an incredible amount of computational time.
Okay, so we've spun the map right side up, fixed the silly putty stretching, and cropped the edges so the boundaries met perfectly. But tissue is messy. Even if the borders match, the cells inside are still full of micro tears and localized warps from being on the lab slide. How do you align the internal cellular streets?
This requires local alignment, and this is the heavy lifter of the framework. CODA applies something called multi-channel LDDMM.
Okay.
That stands for Large Deformation Diffeomorphic Metric Mapping.
Yeah, I'm going to need you to translate diffeomorphic metric mapping into plain English.
Fair enough. Think of it as a mathematical rubber sheet. CODA constructs a vector velocity field.
Okay.
What that means is it calculates the exact trajectory needed to smoothly and non-linearly warp the tissue coordinates back into their true biological shape, pixel by pixel.
Oh, I see.
It corrects the tiny local tears and microscopic ripples, gently pulling the individual cells precisely into alignment without breaking the overall continuous structure of the tissue.
Okay, another pushback, though. If you use a highly advanced, stretchy rubber sheet algorithm, couldn't you just accidentally warp 2 totally different tissues to look identical?
What do you mean?
Like, if I stretch my map of New York hard enough and pinch it in the right places, I could probably make it fit over a map of London. Aren't we risking creating fake biological data just to make the algorithm happy?
This raises a profoundly important question. Overfitting is a huge danger in computational biology, but the developers anticipated this. CODA has a brilliant built-in safety mechanism, a gain-based quality control gate.
A gate? How does that work?
CODA performs that rubber sheet stretch in the background, but before it applies it, it mathematically checks its work. It calculates whether this local stretchy alignment actually improved something called nearest neighbor label consistency.
Meaning, did cells of the same type actually end up next to each other where they belong?
Exactly. If the warp was accurate, similar biological structures should align. CODA requires a specific mathematical improvement threshold, represented by the Greek letter tau, set at 0.02.
And if it doesn't meet that?
If the stretchy local alignment doesn't improve the consistency by at least that 0.02 threshold, CODA completely rejects it. It falls back to the rigid global alignment. It refuses to overwarp the data into a false positive.
I love that. It has a built-in reality check. So let's do a quick recap of this first major concept for you, the listener. CODA is a highly controlled three-step dance.
Right.
First, global alignment to rotate and fix the directional stretching. Second, it turns the data into a color image so a visual AI can crop out the matching common domain. And third, it uses a smart, stretchy rubber sheet to fix micro tears, but only if it passes a strict quality control date to prevent fake data.
Precisely, that's the engine.
So I want you to think about a time you tried to stitch together a panorama photo on your phone, but someone walked through the frame and your phone stitched half a torso to a tree.
We've all been there.
Right. It failed because the software didn't understand the context of what it was stretching. Now imagine how much harder that is when the landscape itself is stretching, tearing, and changing at a microscopic cellular level. That is the hurdle CODA is clearing.
And clearing that hurdle is what opens the door to the real discoveries. If we connect this to the bigger picture, now that CODA has perfectly aligned these tissue slices, we can finally ask, what new biological secrets can it see?
And the answer to that lies in understanding the difference between SCGs and SDGs. Let's define those for the listener.
Sure. So SCGs are spatially consistent genes. These are genes that stay in the exact same physical locations across different samples.
Okay.
You can think of them as your structural markers. You expect to find them forming the scaffolding of the tissue, whether that tissue is perfectly healthy or deeply diseased.
They are the concrete buildings in our city map. They just don't move.
Right. But the real stars of the show, the ones researchers are desperately hunting for, are the SDGs, spatially differential genes.
And what makes them differential?
SDGs are genes where the total volume of expression doesn't necessarily change much at all, but their physical location shifts drastically between a healthy state and a diseased state.
And the source material provided an amazing real-world medical case study for this, looking at atherosclerosis, which is a severe form of coronary artery disease.
Yes.
Researchers compared a slice of a healthy artery to a slice of an atherosclerotic artery.
And this perfectly highlights the flaw in older methods. When researchers use traditional differential gene expression analysis, specifically a common statistical tool called the Wilcox and Ransom test, they look at specific genes like CCL2 and ACE.
And what did the old test show?
Well, the Wilcoxon test basically just looks at overall volume, and it found almost zero statistically significant difference in the total expression volume of those genes between the healthy and diseased hearts. The overall counts were almost exactly the same.
Okay, let me paint a picture for you to really make this stick. Imagine that city map again. The genes are police officers. In a perfectly healthy city, you have 1,000 police officers evenly distributed across all the neighborhoods doing standard patrols. The mayor looks at a spreadsheet showing the total count 1,000 officers. Everything is fine.
Right, a stable environment.
Now look at the diseased city. A major bridge has collapsed. It's a disaster zone. All 1,000 officers abandon their normal suburban patrols and rush to form a massive, dense blockade right at the damaged bridge.
Oh, I see where you're going with this.
If the mayor uses the old spreadsheet tools, the Wilcoxon test, just counting the total number of officers in the city limits, they still see 1,000. They think the city is perfectly fine.
Yeah.
But CODA doesn't look at a spreadsheet. CODA looks at the map. CODA sees that the suburbs are totally empty, and there is a massive localized aggregation of officers and one specific structural failure.
That is a phenomenal analogy, because that is exactly what happened biologically.
Yeah.
When CODA perfectly aligned the healthy and diseased artery slices, it used a spatial cross-correlation metric.
And what did it find?
It spotted that genes like CCL2, ACE, QKI, and HCLS1 were aggregating heavily right around the damaged arterial wall. Their spatial organization had completely reorganized in response to the disease to try and manage the local inflammation.
Wow.
There's a massive biological red flag that was completely invisible to the older volume-based tools.
Because the older tools only saw that the total volume of CCL2 was the same, they totally missed the emergency response.
Exactly. Finding an SDG isn't about counting, it's about mapping behavior.
So a quick recap of the second section for you. CODA finds hidden disease markers by hunting for SDGs genes that physically move to new locations in a diseased state, even if their total numbers stay the same. It catches the police officers swarming the broken bridge. I want you to really think about the medical implications of this. Imagine you were an oncologist designing a targeted cancer therapy. If traditional tools tell you, oh, there are 10,000 cancer-fighting proteins present in this tissue, you might think the patient's immune system is handling it.
But if CODA shows you a map.
Exactly. And you see all 10,000 of those proteins are stuck on the wrong side of a cellular barrier, entirely walled off from the tumor. Your entire treatment plan has to change immediately. You don't need more proteins. You need a drug that breaks down that barrier.
It shifts medicine from a game of quantities to a game of tactical geography. But, as incredible as that is, finding these hidden genes is only practical if the algorithm can actually be run in a normal lab.
Right. Rendering millions of cells in spatial coordinates must create incredibly heavy data files. Does this require a NASA supercomputer, or can the standard hospital use it?
That's the million-dollar question. How does CODA stack up against the competition when it comes to raw performance? The benchmark testing was, frankly, a dominant showing for CODA.
Who are they testing against?
The researchers tested it against current competitors with names like PASTE, Santo, SCGs, and Spatial. To push the limits, they used a massive MERFISH mouse brain data set. MERFISH captures data at single-cell resolution, so we are talking about an astronomical amount of data points,
Right.
over 100,000 individual cells per slice.
So what happened when they fed that massive data set into the older tools?
The other tools literally crashed the computers.
Are you serious?
Yeah, any machine with a standard 32 gigabytes of RAM simply ran out of memory and failed. And even get the competitor tools to run without crashing, the researchers had to severely downsize the data set, cutting it down from 100,000 to just 10,000 cells.
Wow.
And even then, on the tiny subset, tools like PASTE and Stellar left residual angular errors, and SCGs took almost an hour and a half to process it.
Wait, so tools designed specifically for spatial transcriptomics are fundamentally incompatible with the actual size of modern single-cell datasets. They crash standard lab computers.
Pretty much.
That seems like a massive bottleneck for the whole industry. How did CODA handle the exact same data?
Well, CODA aligned that same 10,000 cell subset in 12.4 seconds.
12.4 seconds.
And it did it using only 113 megabytes of memory.
113 megabytes. A web browser with 3 tabs open uses more RAM than that.
Yes. And this goes back to what we discussed earlier, CODA's image-based design. Because it converts the complex spatial coordinates into an RGB-style image and uses computer vision AI, it sidesteps the incredibly heavy mathematical coordinate calculations that bog down the other tools. It's elegantly lightweight.
So because it was so light on memory, they didn't have to shrink the data set.
Exactly. They were
Oh.
able to run CODA on the full, unshrunk 100,000 cell data set. It processed the entire thing in just 510 seconds, about 8.5 minutes.
Processing 100,000 cells in 8 minutes on standard RAM completely democratizes this technology. I mean, any standard university lab, any regional hospital could eventually run this. And the sources show CODA is incredibly versatile too. It has this cross-platform ability where it can align a slice taken with 10x Visium directly to a slice taken with MERFISH, even though they have totally different resolutions and cell counts.
It's very robust.
But the real magic for me, the thing that feels like science fiction, is the serial stacking. CODA can take a stack of multiple 2D slices and align them one on top of the other to build a coherent 3D model.
Yes, 3D reconstruction is a major leap forward. By perfectly aligning consecutive slices, CODA vastly improves something called the Spatial Consistency Index, or SCI.
And what does SCI measure?
Think of SCI as a measurement of smoothness. When you stack 2D slices without perfect alignment, the biological structures look jagged and disconnected, like a poorly stacked deck of cards.
Got it.
With CODA, layers of a brain cortex form continuous, properly stratified sheets in 3D.
Which brings us to my absolute favorite example in this entire deep dive, the axolotl.
Ah, yes, the regenerative biology application.
For those who might not know, an axolotl is a type of aquatic salamander that has the almost supernatural ability to regenerate almost any part of its body. If it loses a limb, it grows back. But, incredibly, it can also regenerate its brain.
And researchers used CODA to look at the regenerating brain tissue of an axolotl, specifically a region called the telencephalon, which is the cerebrum, the front part of the brain, just 2 days post-injury.
That's so fast.
Now, imagine the environment of a regenerating brain. It is highly dynamic and chaotic. Tissue is actively moving, dividing, and rebuilding in real time.
It's a microscopic construction site.
Exactly. Despite that chaos, CODA successfully aligned the injured tissue slices into a 3D model. And in doing so, it highlighted highly spatially consistent genes like GFA and VIM.
Okay, unpack those acronyms for us. Why do GFA and VIM matter to a regenerating brain?
So GFA and VIM are well-established genetic markers for reactive astrocytes. Astrocytes are these star-shaped cells in the brain that, during an injury, rush to the site to form a protective scar and release growth factors.
Like the first responders.
Right. By perfectly mapping these specific genes in 3D, CODA allowed researchers to see the exact spatial programs of the axolotl's incredible regenerative healing process as it was happening. It captured the 3D architectural blueprint of regeneration.
That is just stunning. Okay, let's do a quick recap of this final stretch for you. CODA isn't just accurate, it is insanely fast.
Super fast.
By treating genetic data like a visual image, it processes in seconds instead of hours, using virtually no RAM, meaning it won't crash your computer. It can mix and match data from different platforms, and it can stack 2D slices to build perfect 3D biological models, even of regenerating brains.
It really is a powerhouse.
So think about the scale of what we are doing here. We are mapping 3D structures from 2D slices. We are essentially taking a pile of scattered bread slices and perfectly rebuilding the loaf of bread down to the exact position of every crumb.
That's a great way to visualize it.
What other fields of science or engineering could use an algorithm this fast and accurate at recognizing spatial patterns?
The applications for computer vision, satellite topography, and structural engineering are truly vast. But keeping it strictly within biology, if we connect all of this to the bigger picture, we are officially entering an era where medicine isn't just about what is happening in your body. It is fundamentally about where it is happening at a microscopic topological level.
Exactly. So what does this all mean for you, the listener? Tools like CODA ensure we don't miss the forest for the trees. Or, to go back to our earlier analogy, they ensure we don't miss the damaged artery because we were too busy blindly counting the overall number of police officers on a spreadsheet. We can finally see them swarming the bridge.
It is the profound difference between having a list of ingredients and having a detailed map of a living, breathing ecosystem. And I want to leave you with a forward-looking idea from the discussion section of the source material.
Let's hear it.
Right now, CODA aligns spatial transcriptomics, meaning it maps RNA expression. But what happens when we integrate this exact same framework with spatial proteomics, mapping the actual structural proteins, or spatial epigenomics, mapping the DNA accessibility?
Oh, wow. Layering all those maps on top of each other.
Yes, a true multi-omics 3D atlas. Imagine a near future where AI generative models don't just align the tissue slices we happen to have, but actually predict and fill in the missing 3D biological gaps between the slices.
That's wild.
We could watch diseases form, spread, and heal in perfect reconstructed 3D space over time.
Which raises the ultimate question for you to mull over as we wrap up. If we can eventually watch the exact spatial choreography of a single cell turning cancerous or a single axolotl cell regenerating a broken brain, how will that redefine our very definition of what a disease actually is?
It changes it from a static label in a textbook into a dynamic geographic event.
Incredible. Well, that is all for today. Thank you for joining us on this deep dive into the microscopic map of life. We'll catch you next time.